Welcome all to this special event in collaboration with Microbiome Labs and Fullscript. I'm Amy Regan from Fullscript, and I am so glad that you joined us today. I just have a couple of housekeeping notes. Please place all questions you have in the Q&A box. We will be answering them via the chat during this event. Anyone who has any questions after the event can reach out to me at amy.regan, that's amy.regan, at fullscript.com, and I can help get those questions answered. This event is being recorded and will be sent via email to all attendees and registrants. Excellent. Now, let's enjoy this roundtable discussion on pioneering the gut-brain axis support with Dr. Jeffrey Glad, Dr. Alex Keller, and Dr. Kiran Krishnan.
Hello, everybody. Welcome to this discussion, pioneering gut-brain axis support with Microbiome Labs and Fullscript. I'm Dr. Jeff Glad. I'm the Chief Medical Officer at Fullscript, and I'm excited to have Dr. Alex Keller, who's our Medical Director, and Kiran Krishnan, who's with Microbiome Labs, to discuss this very novel product, quite frankly, and the exciting partnership that both Fullscript and Microbiome Labs has in launching this Zenbiome DUAL. So we'll start by doing a little background on each of us, just so everybody kind of understands the background and where we're all coming from. We're going to spend a lot of time hitting Kiran up with questions on this product that's quite unique and gives us a great clinical focus in terms of how you can help your patients specifically in the realm of not just digestive health, but their mental and emotional health and well-being as well. So from my perspective, I'm still a practicing integrative medicine physician in Indiana and have had a consultative practice since 2010. I see patients about 2.5 days a week now, but I'm full-time as the Chief Medical Officer for Fullscript, working with every team at Fullscript on the product in terms of our future, but specifically also on the quality of our catalog, new and unique products like Zenbiome DUAL coming into this catalog, and just how we continue to help the practitioner prescribe and manage healthcare's best supplements. So I'll pass it off to Dr. Keller for a little background on him. And then Kiran, if you want to tell us about you, that would be awesome.
Super. Thank you, Dr. Glad. I'm Dr. Keller. I'm a practicing naturopathic doctor in Ottawa, Canada, going on about 10 years now. And similar to what Dr. Glad does at Fullscript, our team, the Integrative Medical Advisory team, touches many aspects of the company, from education to decision support to product advisory and everything in between. So whereas Dr. Glad is more on the strategic side, I work more on the operational side in managing our integrative medical advisory team. And as a result, the two of us doing this interview with you, Kiran, is really exciting because we do a lot of this internally at Fullscript with our employees and our teammates. We don't do it externally enough. So we're excited to finally welcome you to the Keller and Glad show.
Well, it's an honor to be on. Thank you both so much for having me. It's a very exciting moment for Microbiome Labs in general. I'm one of the co-founders of Microbiome Labs, and my background is training is as a research microbiologist, and then I went into the business world something around 17 years ago with the big focus around understanding the gut microbiome, and then how to utilize bacterial therapy to modulate the microbiome for beneficial health outcomes. Microbiome Labs was founded based on that thinking that we've got a treasure trove of opportunity to find healing modalities through the microbiome. And we can use the power of microbes if we do it right, if we approach the science correctly, and if we know what to look for. We can absolutely utilize the microbiome as a really powerful tool with the modern day physician. MD, we always say MD moving forward will stand for microbiome doctor, I think, and so I think in the near future, and we're here now in many ways, if you don't address the microbiome as part of your practice, you're really leaving out a significant amount of opportunity to heal your patients. So we're honored to be able to bring this kind of technology to the marketplace, and of course, really excited about doing it with Fullscript and Emerson.
No, that's incredible. Yeah. Again, we know, the popularity of the product line that Microbiome Labs has developed. And so tell us about Zenbiome DUAL. I mean, obviously that's why we're here today. Give us some background, first of all, just what is this? And, so the background on how it was developed, and then we'll kind of dig in a little bit deeper as we go.
Yeah, Zenbiome DUAL was developed by our sister company called Precision Biotic. So in 2021, Microbiome Labs, we were acquired by a large Danish biotech company called Novozymes. We decided to partner with Novozymes because Novozymes actually has the largest collection of well-cataloged and characterized probiotic strains in the world. They have over 150,000 strains and over 200 PhD researchers in the biotech center that are looking to develop technologies around these strains. So they were looking at acquisitions and partnerships with companies that are in the space, in the marketplace, really driving therapeutic use of probiotics. And so Microbiome Labs is one of those. Another company is called Precision Biotics. Precision Biotics was based out of Cork, Ireland, founded by a number of researchers that spun off from the University College Cork. And the APC. The APC is a massive research institute based out of Cork, Ireland, with a big focus around the microbiome. And these two strains that are found in Zenbiome DUAL really were derived from the work at the APC. This has now been well over a decade in developing these. What's really interesting about these strains is that the way they were derived is really unique. Compared to how a lot of other probiotic strains are derived, right? So many probiotic strains are derived by looking at stool cultures from healthy individuals. So you start picking up microbes and characterizing them out of stool. These strains are actually derived out of looking at mucosal samples. And the difference there is that mucosal samples provide an insight into strains that are absolutely colonized or engraft well in the human digestive tract, right? Strains that are found in fecal matter could be either colonizing or non-colonizing. They can be completely transient, but still present in the fecal matter. But once you go into the mucosal lining, you're starting to look at the true commensals that have established themselves within the gut microbiome. Now, the way these two strains were identified was they took actually a number of healthy older women, Irish women. These are based out of Cork. These are women that have very strong constitutions, iron guts, if you will, an amazing history of well-balanced mood, no history of anxiety, depression, any of that. And they started looking at microbiome samples from these women to understand what kind of microbes they house that could have an impact on the gut-brain axis. Both of these strains were discovered through that process. And as it turns out, both of these strains have very profound neurological impact in people. So, we're super thankful for those wonderful women that were willing to donate samples to be studied. And then also we've got a tremendous group of researchers that have gone, deep into the weeds to understand what was isolated and how it actually works. So that's where these were derived from.
No.
Go ahead, Alex, please.
I was just going to say again, we're going to touch on some details here, but if you want to do a deeper dive, because we're not going to go too deep today, consult the PLMI workshop series that was started last month and will be continuing this coming month. But we do want to get a bit granular here and understand how some of these factors were derived. So first of all, for anyone who doesn't understand, how are you taking mucosal sampling? I think, again, most people are familiar with stool sampling, but how are you doing that with mucosa?
Yeah, so you actually do it through endoscopy. That's the only effective way to do it. You actually go in and you're actually, peeling off layers of the mucosa, which is perfectly fine. Doesn't cause any harm, of course, because the mucosa can regrow in healthy individuals very, very quickly, typically within 72 hours. So you're peeling off the very top layers of the mucosa itself. When you find commensals that are well engrafted in the lining of the gut, they typically enter in just in the very top layer of the mucosa. Most of the, especially in the mucin 2 layer. So in the GI tract, there's a couple of different distinct layers of the mucosa. You've got the mucin 1, the top layer that's a lot more liquid-like, and then the mucin 2 layer, which is a little bit thicker gel-like structure. If you can penetrate a little bit into the mucin 2 layer, you're really picking up some key commensals. That's where you find well-known keystone bacteria like Akkermansia muciniphila, for example. They're in those layers, and when you pick up parts of those layers, you're really getting a sense of what the true commensal drivers are in these individuals.
That's interesting, Kiran. So, as I'm thinking about that, sort of the practical application to the practicing physician, you're not going to go measure these levels to determine whether the patient, has enough of these particular probiotics or not. So it becomes very much a clinical discussion of, if the clinical symptoms match these concerns, then, hey, let's think about this product, because picking it up in a standard stool test, shouldn't be the way that we decide that.
That's exactly right. Yeah. So, when you think about these kind of microbes, they are such effective tools in modulating certain mechanisms within the body, right? Whether it's inflammatory mechanisms, neurological mechanisms, which will address both, they modulate these mechanisms in a very powerful manner. So at the end of the day, the clinical assessment is really, do these mechanisms need modulation? It's not necessarily the presence or absence of the microbe. You can assume to a certain degree that these types of microbes are not present if those mechanisms are dysfunctional. For example, if they're suffering from IBS-like symptoms or anxiety or depression, these are all effects of the gut-brain axis and indicate a disruption of the gut-brain axis, which means that they likely don't have the presence of these protective species. So you can assume that and then use these therapeutic tools to modulate those mechanisms.
Could you expand a little bit on that? So how does the gut-brain connection actually work as it relates to microbes specifically? What actual function is taking place there?
Yeah, one of the most fascinating things about the gut-brain axis to me is really how we frame it, right? So we call it a gut-brain axis, but the more you dig into the physiology, the more you dig into the communication, it is bidirectional, meaning the gut is communicating with the brain as the brain is communicating with the gut. They really seem like two parts of the same system, right? It's a singular system, and they're just two polar ends of it. And those two systems are contiguous. They're constantly in communication, and they depend on one another quite a bit. So if you think about some of the basic connections, right? We have, of course, the vagus nerve that connects the enteric nervous system, so the neurological system lining the gut, with the central nervous system itself. And we know now that the enteric nervous system has the second highest density of nerve endings, even more so than the spinal cord, and it's only second to the brain itself. So in many ways, people think of it as the other brain. So they're physically connected. We also know that the gut and the brain influence each other through the lymphatic system, and also, of course, the blood system, the circulatory system. Now, the microbes that line the gut have full access to the enteric nervous system, and they can do that. They can modulate the enteric nervous system, and then thereby through the vagus nerve, the central nervous system, by doing things like producing compounds, right? So we know things like GABA, brain-derived neurotropic factor, serotonin, dopamine, all of those things are produced in large part in the gut. We also know that things like, basic nutrients, peptides and enzymes, and of course, absorption and assimilation of nutrients that are important to the brain, all of those come through the gut first as well. And microbes have a way of modulating these nutrients that can have an effect on the central nervous system as well. We also know that the immune system, the gut houses somewhere around 80% of all of the immune sampling tissue, and a lot of how the immune system responds in areas peripheral or distal from the gut is modulated by what's happening in the gut itself. So when you have sampling that occurs in the Peyer's patches, for example, in the small bowel, that sampling gets amplified through the mesenteric lymph nodes, and then, of course, through the rest of the body as well, through the rest of the mucosal system. all the way up to the brain as well. So what's happening in the gut from an inflammatory perspective seems to impact the brain from an inflammatory perspective as well. So I think the more and more we think about it as the two parts of the same system, the more we'll understand and appreciate the importance of targeting the gut in anything that we associate with the brain, right? Whether it's concentration, alterations in mood, cognitive function, or long-term brain health, things like Alzheimer's, Parkinson's, dementia, and so on. One other note to make, and this is an analogy I give when I'm often lecturing about this, is if you think about, where the gut and the brain relationship lies in an individual, right? You could have it on one end where the gut is completely supportive and easily one of the most important things to the brain, right? And that's on one end. And the other end, the gut is incredibly toxic to the brain. And so most people's gut-brain relationship is somewhere on that spectrum. And the gut can become arguably one of the most toxic things to the brain on a regular basis. A leaky gut or heavily dysbiotic gut that produces lots of toxins and inflammation and all that becomes incredibly damaging to the brain over time.
Yeah, so let's all that background is incredible, and gosh, the way you explain it just makes it so clear. I also think about from the integrative practitioner perspective, right? The breadth of other aspects, as you mentioned, stress and sleep and nutrition all become really important too. But let's talk about Zenbiome DUAL, and you've got two specific bifidobacterium species in there. Would love to hear you talk about sort of how we think about the mechanism of action for each of those species and why they were chosen to be in this unique product.
Yeah, absolutely. So they're both Bifidobacterium longum. One is Bifidobacterium longum 1714, and the other is Bifidobacterium longum 35624. So let's tackle the 1714 first. Now, both of them are what we call EPS strains. So these are strains that have an exopolysaccharide coating around them, which is actually quite unique to longum or Bifidobacterium in general. You don't find a lot of Bifidobacterium subspecies in the gut or outside, through probiotics and so on that have this exopolysaccharide coating. So the exopolysaccharide coating is basically a very complex sugar-like carbohydrate coating that is produced by the microbe itself. And you can see it in an electron micrograph. It looks like all of these wonderful little finger-like projections that surround the microbe. And I'll come back to the function of the EPS, the exopolysaccharide, because that's actually what makes these strains function the way they do. Now, what's unique about the exopolysaccharide coating is they're typically coded for by one, maybe two genes, right? So if you take a bifidolongum that doesn't have an EPS, compare it, for example, to the 1714 strain, the 1714 has one gene that provides the EPS functionality that the other Bifidobacterium longum would not have. And that one gene completely separates the functionality of these species. So we've been able to show by knocking out that single gene that codes for the EPS, once you do that, once you knock out that single gene, the strain doesn't have any of the same functionality at all. And we'll talk about what all those functionalities are. But it's all reliant on that EPS gene, which is really, really fascinating, right? So this is where it becomes so important to understand these microbes in a very intricate way and know that there's no replacement for it, right? You might see in another product, Bifidobacterium longum, but if it's not 1714, it means it doesn't have that one gene and it doesn't code for EPS, and that thereby it doesn't have all that functionality. So let's talk about what happens with this strain. So 1714 with the EPS, what happens when you swallow it is it does survive through the gastric system. Most Bifidobacter can't do that. They're relatively sensitive strains, but the EPS coating actually provides a protection to be able to survive through the stomach acid and then, of course, bile salts as well. It migrates its way through the small intestine, and then when it gets to the terminal end of the ileum, what tends to happen is it actually gets phagocytized by dendritic cells. So in the sampling portion of the ileum, the Peyer's patches, dendritic cells have this kind of transmembrane component where they're peeking out into the mucosal or luminal side constantly, looking at microbes and looking at things that are coming into the gut. It recognizes this EPS, and it uses that recognition to grab onto the strain and pull it across the lining of the gut and actually engulfs the strain. Now, here's where it becomes super interesting. Because of the EPS, right, there aren't any antigenic moieties that are structured on this strain, meaning that there's nothing on the surface area of the bacteria that actually creates an amplified and antigen presenting like signaling from the dendritic cell. So what does that mean? Well, normally when dendritic cells engulf a bacteria, what they're doing is they're engulfing the bacteria, and then there are some antigenic sites on the surface of the bacteria that causes the dendritic cell to activate a cascade of inflammatory responses. So it activates interferon gamma and alpha, for example, or TNF. And in the same process, it then processes the bacteria and presents antigens to T cells and B cells, right? Helper T cells. Now, it doesn't do that in the case of 1714. It doesn't present any antigens. It, in fact, engulfs the bacteria, no expression of inflammatory cytokines at all in that process. And during the engulfment, it digests the bacteria, and it releases the carbohydrates from the bacteria, from the EPS, and from the membrane of the bacteria where you find things like peptidoglycan. It releases that into circulation. And that part is the really fascinating part, because at the end of the day, the released carbohydrate from the dendritic cell is what's going to modulate neurological and inflammatory responses in the body. So the way 1714 does is the released carbohydrates, both the peptidoglycan and the digested exopolysaccharide, has a neuro and systemic inflammatory modulation. It does that by significantly increasing IL-10, IL-10 being the anti-inflammatory cytokine in the body. It significantly increases Treg function, so the regulatory T cells that can dampen and modulate unfavorable inflammatory responses. And it specifically brings down things like TNF alpha, interferon gamma, and so on. So it's complete modulation of the mechanics around inflammation that occur in the mucosal lining, and then eventually in the neurological system, and then throughout the body as well. Now, what's interesting about the type of peptidoglycan and the type of carbohydrates that exist on the 1714 strain is that those tend to be tropic or attracted towards the central nervous system. So they have an affinity for moving to the central nervous system and modulating neurological activity or inflammation in the CNS itself, right? So that's where we see stress response being heightened by elevation of inflammation in the CNS. And then these carbohydrates from this unique bacteria come along and downregulate all of those inflammatory responses, thereby reducing the impact that stress has on the individual. And it even reduces the perception of the stress by modulating the inflammatory response. There's another functionality which we don't quite understand the mechanism of action yet, but the 1714 strain also seems to change brainwave activity. This has now been well documented and published by Professor Paul Enck out of Germany, where he shows that when you have an individual that is susceptible to stress and anxiety response, when you put them in a stress state, they tend to turn on high frequency brainwaves, right? So they tend to turn on things like gamma waves and beta waves at a high frequency. When you turn on high frequency brain waves, what tends to happen is your brain actually creates a higher perception of that stress, and it puts you into a spiral in a more profound manner, versus reducing the high frequency brain waves and turning on the low frequency brain waves. In this case, turning on things like alpha waves, right? And the microbe has a capability of changing brainwave function when you respond to stress, thereby dramatically reducing the perception of the stress, and also increasing the activity of the coping centers of the brain, which helps you then rationalize through the stress better. right? So think of 1714 as central nervous system, reducing inflammation, reducing cortisol curve as a result of that, and then also changing brainwave function to modulate anxious and stress response in individuals. So that's the first half of it. And so 35624 works in a very similar manner. Now, 35624 is also an exopolysaccharide strain. It also has that EPS layer. It also has peptidoglycans within its cell membrane. And it works in a very similar way in which it is attracted to dendritic cells. Dendritic cells do engulf it and digest its polysaccharides and release its polysaccharides. But it seems like 35624 seems to have more of an inflammatory modulation in the enteric nervous system, and to some degree in the central nervous system, but more so in the enteric nervous system, where all of these hyperactive neurological signals are coming in from the brain that is causing hyperactivity of the enteric nervous system, which thereby causes these IBS-like symptoms, right? Too much peristaltic activity, overactive movement in the bowel, creating gas and bloating and all the characteristic symptoms associated with IBS. That is all inflammation mediated as well in the enteric nervous system. 35624 seems to modulate that in the enteric nervous system, similar to how 1714 does it in the central nervous system.
Kiran, that was an incredible explanation. I'm sitting here thinking, I wish you had taught my immunology class.
Immunology is one of those crazy areas where, unless you make real sense of it, right, it's just so hard to grasp because the mechanisms behind them are so profound.
Yeah, absolutely interconnected in how it's all functioning. The concept of digestion of the microbe to release the carbohydrate or to release the polypeptide, that is not something I've heard before. I mean, to think that the destruction of the microbe is actually the therapeutic in itself. Yeah. Just out of curiosity, why or has there been any effort to isolate the actual carbohydrate or the polypeptide instead of doing it through this kind of mechanism?
Yeah, there is. So actually, we've been working on a project where we're looking at a couple of different mechanisms. So one is you take the strain itself, but with a enzyme that actually, which is a lysozyme, if you will. You take it with it, which actually then in a certain part of the GI tract, where the lysozymes are pH sensitive. So in a certain part of the GI tract, predominantly in the distal part of the small intestine, the lysozyme then starts lysing and breaking up the bacteria. And when it does that, it releases more of the carbohydrate, which then presumably more of that carbohydrate gets absorbed and then becomes available to the circulatory system to modulate inflammation. We've done some preliminary studies on that in animal models, and we've seen that can actually enhance the effect even more. We've also looked at isolating the exopolysaccharide itself and producing that at higher levels. That is hard to do because the yield of it is really low, because it's not a excretory compound that the bacteria produces, right? It produces a certain amount of it to coat itself, and as long as it has a decent level of coating, it maintains that level. It doesn't continuously produce it and excrete it like microbes do with enzymes and vitamins and so on. So it becomes hard to generate a high yield version of that. So at the end of the day, the next evolution may be the combination of this strain with the lysozyme, which could enhance the exposure of these carbohydrates into the system. Now, here's another really interesting insight around that idea of dendritic cells somehow recognizing microbes that have exopolysaccharides and these unique carbohydrates and engulfing them and digesting them, right? This also happens in utero. In utero is really interesting. And this is where these EPS and peptidoglycan, which are both found in these strains, becomes really foundational to brain health, because studies have shown that the placenta contains peptidoglycan binders and peptidoglycan carriers, and bacterial peptidoglycan, that is. And then the baby's brain in utero has receptors for bacterial peptidoglycan. Then the question comes, well, how is it that the placenta and the baby become exposed to bacterial peptidoglycan, given that many of those microbes are on the other side of the mucosal lining in mom's gut? As it turns out, there's some evidence that dendritic cells in mom's gut specifically go across the membrane, look for these types of bacteria, grab them, bring them across, digest the peptidoglycan and the carbohydrates, and release it into circulation so they make their way to the placenta. And in the placenta, we have a receptor to bind bacterial peptidoglycan, then a carrier that carries it to the baby's brain. It binds in the baby's brain. And then when it binds in the baby's brain, the things that it stimulates are mind-boggling. It stimulates differentiation of different parts of the brain as the baby is developing. It stimulates synaptogenesis. So it increases the number of synaptic regions that the baby is developing in utero. It increases the development of the blood-brain barrier and also myelination of the central nervous system, right? Arguably, some of the most important things we want to happen in the brains of our babies as they're developing in utero, we've outsourced that to bacteria. And so these exopolysaccharide peptidoglycan microbes have been important for your brain from day one in utero. And as it turns out, they're important in adulthood as well to modulate the negative effects of stress and inflammation on the brain.
Wow. So, I want to move a little bit on here, although, I have so many questions, but I suspect a lot of this may be covered in the PLMI 3-part series that's happening as well. So if anyone wants those, deep dives and better understanding of the gut-brain axis and how this all fits in, please go there. I think the third one will be next week, but I think you'll be able to find the other two. Alex, from a protocol perspective, now I want to get into sort of the practical application of how this fits into practice, right? Can you talk a little bit about what you and your team have developed from the Microbiome Labs gut-brain axis protocol that practitioners can, easily bring into their recommendation and prescribing engine within the platform?
Yeah, so we've developed in partnership with Microbiome Labs a protocol that you can apply directly on Fullscript that includes numerous products and recommendations. One click, super easy to apply. We're going to include it in the email follow-up after this webinar and in the show notes so that you can go and access it right away. But the beauty with this application, and I'll mention what the products are here in 1 second, but you can, with one click, apply this to your Fullscript platform or page and be able to prescribe it right away. Very easy to apply. So the products, and maybe, Kiran, you can expand a little bit on why we would combine these products together, include Zenbiome DUAL, Megaspore Biotic, Mega IgG 2000, Mega Marine, and then for a longer period, FODMAPE. I should say that those first products are specifically indicated, I believe, for 1 week, and then you extend from there, periodically different either dosages or you use the FODMAPE over the long term. So, Kiran, can you expand a little bit on why the combination of those products and why there's mutual benefit?
Yeah, absolutely. So when you look at those combinations, and this is such a powerful protocol and easy to use, the combination really attacks many of the pathologies that drive gut-brain dysfunctions, right? And we're talking about primary GI issues like IBS, as well as anxiety, mood disorders, sleep disorders, and so on. So we talked about the Zenbiome DUAL, and we know what that does in terms of modulation of neuroinflammation in the gut, in the central nervous system, changing of brainwave activities, and also increasing the diversity of the gut microbiome. We do have some data that it does modulate the gut microbiome as well. The Megaspore becomes critically important in this whole process because one of the key culprits that drives a significant amount of brain inflammation and central nervous system inflammation is lipopolysaccharide. So LPS from gram-negative bacteria. having elevated levels of LPS in circulation is the biggest risk factor for the development of anxiety, depression, and long-term neurodegenerative conditions, right? So this has been shown in Alzheimer's studies and Parkinson's studies. The big Netherlands anxiety and depression study, which was a 9-year study on over almost 3,000 people, showed that the biggest predictor of the risk of development of anxiety and depression and neurological symptoms is elevated levels of LPS, lipopolysaccharide, which is an endotoxin that's made by gram-negative bacteria in the gut microbiome. And at least half of the microbes in your gut microbiome are gram-negative bacteria. So it's an endotoxin that's constantly present in your gut. Now, if your gut is leaky, if you have intestinal permeability, that endotoxin is allowed to leak through, creating a condition called endotoxemia, having elevated levels of LPS. So Megaspore was the first product shown starting in 2017 from a publication in World Journal of Gastrointestinal Pathophysiology that taking the spore-based probiotic dramatically reduces LPS endotoxemia, right? So if you have LPS as a constant driver in your patients, if your patients constantly have leaky gut, as much as the Zenbiome DUAL will help, you'll continue to stoke that inflammatory fire in the central nervous system and the enteric nervous system as well. So it becomes really important to stop the influx of LPS, and that's where Megaspore comes in. Now, the Mega Marine, the high EPA fish oil, is really interesting because when you look at the impact that LPS has on the inflammatory response in the body, it's an inflammatory response that's driven by NF-kappa B and TNF-alpha, then eventually IL-6, IL-12, and so on, right? So that's one inflammatory cascade. There's a second thing that's happening in the gut that translates to the rest of the body, and that's through the prostaglandin system, right? So this is the lipooxygenase and cyclooxygenase pathways. That can also lead to systemic inflammation. And in fact, one of the really strong markers, and this is something that every healthcare practitioner can check with their patients, is something called the AA-EPA ratio, the arachidonic acid to EPA ratio, EPA being the omega-3 fatty acid, right? So when you have high levels of arachidonic acid relative to EPA, what you tend to have is a lot of differentiation of arachidonic acid into lipooxygenase and cyclooxygenase, which means a lot of inflammation in the lining of the gut that translates to inflammation in circulation as well. So lots of conditions like cardiovascular disease, aging, peripheral vascular disease, and lots of other inflammatory conditions are driven by that elevated AA-EPA ratio. When you increase EPA levels in the body, in the gut especially, you bring down that ratio and it modulates that inflammatory pathology. So that becomes a way of attacking this second cascade of inflammation. Remember, the microbes, the Megaspore, the Zenbiome DUAL, the 17, 14, 3, 5, 6 to 4 strains go after the NF-kappa B, TNF-alpha, IL-6 pathways. This one goes after the arachidonic acid pathway. So when you can address both of them, you're really shutting down the vast majority of inflammatory pathways that stem from the gut that impact the complete system in the body, right? The FODMate is a fascinating product because a lot of people with IBS have SIBO-like symptoms. And so what they tend to do is they tend to eliminate FODMAPs, right? These are any sort of fermentable carbohydrates foods. Most of them are very good, healthy foods, but they simply don't tolerate them. And the problem with eliminating those is there are components of those foods that are really important prebiotics for the really important commensal bacteria in your large bowel. So people with these symptoms tend to avoid those foods. They tend to concentrate on nothing but protein and fats, and they're not feeding the important commensal bacteria in the large bowel that are the ones that are making things like butyrate, short chain fatty acids, and so on that are important for gut function. FODMate is a unique enzyme blend that allows you to tolerate FODMAPs, which are these fermentable carbohydrate foods that allows you to diversify your diet, feed those important commensals that are really important for modulating inflammatory response in the mucosa, also producing things like butyrate, propionate, and acetate, and then allowing you to diversify your microbiome, which at the end of the day is one of the most important markers of a healthy microbiome is that alpha diversity in the microbiome. So FODMate allows you to do that, so you don't have to go through this really restrictive elimination diet. So you add all those components together, and you've got a very nice comprehensive approach to patients that have intestinal permeability, neurological inflammation, and all the associated symptoms.
Love it. Yeah, that's incredible. So we've got about 10 minutes. I want to make sure we cover everything that we've got here on our list. I'll ask one quick practical question. Alex has got a question on sort of understanding the quality and safety story that you guys are really focused on at Microbiome Labs. But just practically, I hear, depending on the conference, the company, the product, how should I take my probiotic, right? And particularly Zenbiome DUAL. Is this with breakfast? Is this a empty stomach? What's the data tell us in terms of dosing this?
Can I plug in there quickly and also ask, when would you take Zenbiome DUAL by itself versus applying this protocol?
Yeah, absolutely. So so you would take it. You can actually take it with or without a meal. In this case, because really what we want to do is we want to get the strain down to the Peyer's patches where it'll get sampled by the dendritic cells. And the presence of food is not a problem for it because it is acid stable. A lot of probiotics that are not acid stable, we're trying to skirt around the release of HCl when you start eating food, and thereby recommending taking it in between meals. So in this case, you could take it when you remember during the day. If you end up taking it with food, that's absolutely fine. I end up taking it before my first meal because there's a certain number of supplements I take right before eating, and it's just easy for me to remember. So that's when I take it in the morning, which is, well, actually, it's not in the morning. For me, it's late morning because I'm typically fasting through the morning anyway. Now, I always take my Megaspore, for example, with the meal. Right. So we've found that the spores tend to have higher levels of activity when it's coming in with food substances. And that matches what happens in nature. Normally you're getting spores by inadvertent exposure as you're eating, roots and tubers and things that you forage and gather in the environment. And the spores require the presence of some amino acids and some carbohydrates in order to come out of the spore form and become a vegetative functioning cell. So I always take Megaspore with the meal, and then I always take the Zenbiome DUAL right before my first meal of the day.
Super. As Jeff said, we're coming up on time here, so we're going to start to wrap it up, but we do want to talk about quality because quality is such an important part of the healthcare provider ecosystem and the products that we want to make sure that our patients are taking. This is near and dear to our heart at Fullscript with regards to patients being able to purchase products all over the place these days and wanting to make sure that they have a trusted source for products. In the case of Microbiome Labs, and in this product in particular, can you speak to the manufacturing standards that you apply and how, in this case, when you're working with multiple partners, you're still ensuring that the quality standards are top-notch?
Yeah, absolutely. That's another really beneficial part about being a component of Novozymes. Novozymes is a multinational company, biotech company, highly focused on quality and using the highest standards of CGMP, right? So that's where it starts. It starts with the manufacturing process, and we own the manufacturing process from the very beginning. And that's another way of ensuring quality is that we own everything from soup to nuts. We own the strains, the original culture. We own the manufacturing plants. We produce the product. We isolate the product. We study the product. And then we even finish the product in capsules and bottles, which means that we control the process in every step. And we're not dependent on third-party manufacturers for that. When it comes to strain quality, ensuring quality among probiotics, the first step is proper characterization of the strain, right? We need to know what this species is inside and out, which means full genome blasting and looking at every single protein and every single thing that the microbe can possibly produce. For that, you need a very sophisticated data science and genetics team. At Novozymes, we've got an entire team that all they do all day long is genetic analysis on microbes, right? So first, it starts with characterizing the strain properly, understanding every functioning gene in the microbe so that we can understand, does it produce anything that may be egregious? Does it have any chance of producing a toxin, right, that could compromise health and safety? And so all of that happens in the background. And then during the manufacturing process, there are steps in which you keep sampling the manufacturing culture and analyzing it genetically to ensure that no genetic changes have happened with the strains, right? That's a very important thing because, microbes can have mutations, random mutations, and we want to ensure that the microbe we seeded the fermentation culture with is the exact same microbe that comes out on the other side. So you need these genetic checks through the process of fermentation. Then once the microbe comes out, it's all about recharacterizing it, making sure it's exactly the same microbe that we intended to produce. And then we only produce these microbes in isolation. We don't mix microbes during fermentation and culture, because when you mix microbes, they start to share genes, right? And we don't want that to happen because we want to be able to control the genetic elements of the microbes. And then, of course, isolating the microbes, concentrating it, spray drying it, and then doing every possible check that you can do on that finished powder. Everything from pesticides, lead, mercury, pathogens, all of these things are checked well beyond CGMP standards. And then bringing that microbe and then having the highest standards in encapsulation, bottling, and so on. Now, the other aspect of quality is also the research that needs to be done, right? Because what we want to understand not only about the microbe itself, but how does that microbe play with the rest of the microbiome? That's a very important component that is often not checked by probiotic producers, because when you put a microbe into your system, not only is it the effect of that microbe that has an impact on you, it is what does that microbe influence the rest of the microbiome to do, right? So that has to be checked as well, and that's something that we check before we ever release a product. So all of these checks combined together becomes a passport for the bacteria, and that passport has to be exactly the same every single time we produce them.
I think everyone can appreciate, how much goes into the quality story and why Microbiome Labs is such a great partner for Fullscript in our catalog. So I want to move on to just a few questions here that we've gotten in here as we've gone. This one's a practical one. Kiran, tell us about children and Zenbiome DUAL, and what are your thoughts? What maybe the evidence suggests, or how should practitioners think about that as we talk about children who may need this, specifically with mood concerns?
Yeah, absolutely. So the good thing about these strains is the way they work is it's your own immune system that utilizes the strains in a way that it modulates inflammatory response. It doesn't cause any sort of Herxheimer reaction or die-off reaction. So there's no issue of sensitivities of certain patients and some patients having this die-off response and so on. So it's absolutely applicable for children as well. My kids, my 9-year-old and 12-year-old, both take the Zenbiome product because, our kids are inundated with pro-inflammatory stimuli in the environment that they live in, and the foods that they consume and so on. So for me, modulating their neuroinflammation becomes really important. And you see it in the psychology of kids, right? We know that ADD, ADHD, anxiety, depression, and all that are at epidemic levels among kids. And a lot of that is due to that neuroinflammation and the diminishing quality of the microbiome. So I utilize these with my kids. We do have a couple studies in younger populations under the age of 12, and there's no toxicity issue at all, right? These are absolutely commensal organisms that are very, very safe. So there should be no concern about utilizing them with kids. Of course, these are coming in capsule form, right? So my 12-year-old's finally gotten around to the idea of swallowing capsules, but it took a long, long time to be able to do that. My 9-year-old doesn't. So in her case, we open it up and we mix it in yogurt. It's not the most ideal delivery mechanism. It's great to do it in a capsule, but it's absolutely still beneficial when you do that. So with kids, mix it in some sort of soft food and have them consume it immediately. You don't want it to sit in the soft food for a long time because the strains aren't stable in that kind of environment for a period of time, but mix it in, have them take it right away.
That's perfect.
Super. All right, Kiran, we're going to wrap it up because we're at time, but I'm sure there will be follow-up questions. And as always, we can field those questions and answer them after the fact if we need to. We also mentioned that we're going to share a number of notes and resources in the show notes and via email after. So there will be more information coming. I wish we could spend 3 hours with you or more. So it'd be ideal to have you back another time. We can expand on this if we want to. But in the meantime, we do want to highlight that there is a discount right now that can be applied on Emerson using the promo code MBL10, where you get a 10% discount off Zenbiome DUAL. So make sure that if you're looking to purchase this product for your clinic or otherwise, use Emerson and use this code for a limited time. And also to mention that we will be launching a course on Fullscript Academy all about this, where we'll dive into more details. And again, to mention one more time, that if you want a real deep dive, tune into the 3-part series that PLMI is hosting at no cost. Lots of valuable information in there. But for now, we'll say thank you very much and see you again soon, because we hope to have you back on the Keller and Glad show.
Yeah, thank you. It was an absolute pleasure. And I want to make one final statement. This is very exciting for us to be able to do it. Just keep in mind, it's almost 2 decades worth of research that has gone into developing these strains and understanding how they function. But none of that would be meaningful at all unless it was accessible to people. And we're extremely grateful to Fullscript to be able to do that for us, where in this partnership, you make it accessible to healthcare practitioners that are highly conscious of going after root cause medicine. It's estimated that around 15% of the global population has IBS, and almost 90% of people with IBS have anxiety. And even though IBS and anxiety are things that people kind of live with and manage through, what's important to note is that the underlying pathologies of those 2 conditions are also the underlying pathologies of many long-term disruptions, right? Things that are really scary, things like dementia, Alzheimer's, and so on. And so addressing those pathologies now, when they're showing up as IBS and anxiety, can be extremely powerful and beneficial. And we're very grateful for the partnership with Fullscript to get this product out there to the doctors.
Gosh, I'm not sure neither Dr. Keller nor I could have wrapped that up much better. So yeah, I appreciate the partnership, appreciate your expertise, and I look forward to the future and where this partnership goes. So thanks, Kiran.
Thank you.
Alex, good to hang.
You too.